first of all, thank you very much for this very lively discussion during the talk of Chris Beecher, because I think this is what conference should be about. And for me personally, I benefited a lot from these general fundamental things. So I will try to show you in the next minutes 2 examples how we can use the stable isotope labeling approach to follow the metabolization of tracer compounds in an untargeted way. So I will take one example with a mycotoxin that is working here. Looks good. So for the mycotoxin and for phenylalanine, which was applied to grapevine berries, and we wanted to study how they are metabolized by the berries under different conditions. And if there is a few minutes left, I could also show you how the software performs for maybe one data file. Then you can see how the data are treated and what a typical outcome can be. So I showed this slide in the morning already, and for now I would like to focus on this tracer metabolization thing. And so what you can expect from such an experiment is depending on what type of metabolite you're going to apply. So if you think of a xenobiotic, like the study was mentioned before, if you treat plants with herbicides, for example, or also with a mycotoxin would be the same case. Usually what you observe is that you get a conjugation product. So the detoxification by the plant leads to a conjugate, which is has a higher mass, and also the conjugated moiety will leave will contribute to the isotopic signature of what you can measure. If you choose the right endogenous metabolites, like aromatic amino acid, for example, you can observe a similar thing. But if you imagine here, you apply 1-in-1, a 1-to-1 mixture, then you immediately imply, so if the metabolism is comparable for the C12 and for the C13 material, for the same compound, then all your metabolites will carry will also have the same intensity. All the conjugates, the C12 and the C13 pendant of that, will have the same intensity. If you apply a tracer to as an endogenous metabolite, then you cannot predict this anymore. So you have to consider that this ratio was not known, like in this controlled way, but you have to just see what you can measure. If you take an endogenous metabolite which is highly metabolized, like in the central metabolism, then usually you do not end up with different separated isotopic clusters, but you see that the individual isotopes are contribute to a more complex pattern, which is only one. So I will stick to these first 2 examples here. This is just to make us a little bit modest as the metabolomics community. These concepts are very old, and this I was becoming aware of that by reading through this review article here, which I can strongly recommend to you, which was written in 1980 already, 81, and basically describes where this technique with the ion doublet or double ion or twin ion, whatever you will call it, really comes from. So all was about GCMS in the beginning, and looking for the traces, typical isotopic signature of chlorine, which is very characteristic, was the key to develop this. And this was first, I think, demonstrated by this author here, and then a year later it was also used for to study the metabolization products of barbital compound with a nitrogen labeled compounds, and it all follows the same principle. You try to detect these ion doublets or ion clusters, isotope clusters, whatever you call them. So this was well established in the early of the 70s already. But the difference now is that we have the tools to do an automated querying of the data and to get a fast answer which is comprehensive. So what is the workflow about? I already mentioned you could use a xenobiotic, prepare a mixture 1-in-1, for example, and apply this to the biological system. Or you could use an endogenous tracer and only apply the labeled compound. Then you do your sample preparation, do the measurement, and the concept of data processing is then finding these doublet peaks again. And in this case, when you know something about how the metabolization can be expected and you have a defined ratio which you applied, you also can assume that this has to be in a certain relative intensity which you can use. Then you do the peak picking and peak matching again, and all the features which have their counterpart in the C13 side, they are relevant and are kept. Then de-isotoping step, which is also mentioned again, and all the features are grouped again, and then you have all the data which remain in the after cleaning and correspond then to potential metabolization products. So the first study with the mycotoxin deoxynivalenol, we wanted to see which metabolization products can we measure if we apply it to wheat. So a little bit about the biological function of this compound is produced by a fungal species called Fusarium, and Fusarium graminearum is producing this compound as a virulence factor. And this general stress response by the plant as a response to the presence of the fungus is that polyamines, for example, are induced. And this is a signal which the fungus recognizes and uses for the induction of the production of the toxin. And once the toxin is produced, it's causing oxidative stress and trying to force the cell to death, because Fusarium is basically a hemi-necrotroph, so it has a biotrophic phase in the beginning, and then it's killing the plant cell, and this is the basis that it can really grow inside the plant. So what is the response of the plant? The plant, what was known before we did this study, can conjugate deoxynivalenol to glucose. And this has also been associated to the level of stress response to the fungus. And so we were interested if there are more than these metabolites which are produced as if you apply the mycotoxin directly to the plant. Again, to remind you of what you can use analytically, you have the toxin which you apply as 15 carbon atoms, and if a metabolization, typically a conjugation reaction occurs, then you have an increase in mass, so the whole pattern will be shifted to higher M over Z values, and this can directly be used to annotate the conjugate then, because the mass shift directly tells you the mass of the conjugated moiety. Then the isotopic pattern, of course, can be used on this side also to get an idea of what could be conjugated, and it helps you to identify this as a conjugate. The mass difference between the principal ion tells you how many carbon atoms of the tracer are still in the metabolization product, and the intensity ratio, we already discussed this, depends on the study design. If you know it, you can use it to find these things. So this is a short summary of what the data look like if you apply a mixture 1+1 of non-labeled and fully labeled DON. This is the raw data, how they look like, and if you process it with MetExtract, I hope I can show you afterwards how the data look like, then it's basically clean. It's just showing the metabolization products. Of course, you have to go through these features manually again, could also always happen that the one or other could occasionally be false positive or could be wrongly grouped or something like this. So this has to be verified in any case. This is how the chromatograms look like, and here you see on the positive, and so pointing to the top, the C12 monoisotopic C12 features of all these, which have been automatically annotated, and downwards, this is the C13. And you see it's perfect coelution, and the ratio 1:1 is directly visible in the data. So what we did then is we took all these peaks and did MS on them, and we were able to annotate the structures of those. So what you can see is that it's principally two modes of detoxification. One is via glucose-related metabolites, and the other one is via glutathione conjugates. And you see the intact glutathione and also the degradation product of this of products of this glutathione conjugate in the following. So to summarize, what could be the mode of action is the fungus invading the plant is using the plant signals to induce the production of the mycotoxin. This is then most probably in the cytosol conjugated to glucose and glutathione and actively transported into the vacuole where it's further metabolized. There's two metabolites left which we do not know what it is. One is also a glutathione conjugate, but it's not straightforward to, so far we do not know exactly. And there's another one we are working on to really nail down the structure. So second example, as I indicated already, would be about aromatic amino acid applied to grapeberries under heat stress. And the biological study was carried out at the University of Davis by Alex Chassi, who was formerly was a PhD student in Andrew Waterhouse lab. And Alex is now working in the Fian lab as a postdoc. He's also now into metabolomics, maybe because our data looked so fascinating, I don't know. So we got in touch during a conference, and then we talked, okay, we could just have a look at your data, see if we can find out something, and that worked very well, as I would like to show you now. So the story behind is that I was told, I was once in California, I would like to go again, but in summer it can be very hot. And these high temperatures cause problems for the wine growers, because these high temperatures are a reason for a degradation of the anthocyanins in the grapeberries, and then it is not enough color left for this good Cabernet Sauvignon at the end. So this is a problem, and they wanted to look into the molecular mechanism. So what is behind that, or could we maybe identify some metabolites which are directly linked to that? So the study was as follows: the grapeberries, the grapes were harvested at Viraison. That's when they start to change the color from green to red. Then the metabolism is changing, because then it's not the sugar going. So then the berry starts to produce sugar itself, and it starts to produce the pigments, the colored pigments. So, and then it was the berries were cut in half, and the two halves were then one half was put at 25 degrees centigrade, and the other half was put on 45 degrees centigrade to simulate this heat stress. And this was done in 8 groups with 4 berries per group, and this is what has been measured then afterwards. This is how the data look like. In this case, we were looking for M+6 features, and here you see part of the chromatogram just to illustrate how how typical the pattern looks like. And on top of this M+6, we also found accidentally, we did not look on purpose, but we found also M+12, so indicating that's 2 of the aromatic rings originating from the amino acid have been incorporated into metabolites, which are now visible here. A closer look then, an overview, as an overview, I can tell you we found 112 feature pairs, which have been carrying this signature by this labeled phenylalanine, and 23 of those could be metabolically assigned to either being a M+H, M+sodium, or M+ammonium ion. These corresponded to 63 metabolites, and they corresponded to hydroxycinnamic acids, anthocyanins, flavonols, and stilbenes. So also resveratrol, for example, was found. And this is just to summarize and to indicate that this is the groups which have been differentially affected. So the metabolites which really showed different concentration level due to the induced heat stress. So, and this was evaluated with a paired t-test. So we used the pre-information from the study design that we measured the very same berries which have been split in half. So we only were relating the difference between the treated and non-treated for the same group, within the same group. And this gives you much more statistical power than if you just average a group and another group. It's much more powerful test. And then the red arrows indicate that these compounds have been induced by the heat treatment. So these are potential degradation products of something which we do not know what it is right now. And the general conclusion was that the anthocyanin levels in general were lowered by the treatment. So as you expect, because the color yield is also low under heat conditions. Also, what we observed is that resveratrol and the resveratrol dimer also changed in the concentration level. So I think this was basically what I would have used to show you the two maybe applications which are typical or can be typical for this approach. So I would now have about maybe 5 minutes or maximum 7-8 minutes to process one data file if you're interested. Otherwise we could skip and just jump to the next speaker. No. You mean in this particular study now? Yeah, now. No, I think they just shrink and get dry. No, yeah, they get sweeter if they lose water. This is used for some wine production, but they're not like bananas, for example, or like mango.
So in your case, does that not inhibit your study to some extent when the change is not going on any further as
you could argue like that, but I think the data tell you the difference, because there is a differential response still in incorporating and metabolizing the phenylalanine, which is added from externally.
Yeah, that's what I mean. What you use that as the sugar content which you taste, not of course secondary metabolite. And the question to the secondary metabolite and the decrease in color in the do you know how much of this could be related just to some general stress which leads to more oxidized species, peroxide and so on, which will destroy the
Yeah, that's a good point. We don't know if it's really enzymatic degradation, could also be a chemical degradation of the maybe reactive oxygen species, which can induce also breakdown of the pigments. Yeah. How we did that? No, that's not an artifact. This is if you extract all the features, then as a matter of fact, the same metabolite can form different adducts and also in-source fragments, and all the things are grouped together. And by the mass difference between which you can, if you pre-assume some potential adducts, you can just inspect the groups if these relationships are present, and then you can assign the type of ion to the individual features. You don't look happy now. No, it's just this is just part of the technique. So this happens. This is the electrospray process. Yields in different ions per metabolite. It's a soft ionization technique. Usually you don't have much in-source fragmentation, but depending on the compound, these groups which we observe can be pretty complex. If I run this example, there is one of the metabolites which you see that it's, I think, 20 or 25 different ions belonging to this one ion, to this one metabolite, which are formed in the ion source. Pardon?
I apologize, I still don't understand why there would be a preference for the heavy label.
There is no preference for the heavy label. Maybe I'd put that a bit not very clear, but this is, you can assume that the non-labeled and the fully labeled, they behave the very same in the mass spectrometer, except that you can separate them by mass over charge, of course. Yeah, but this is the reason why you can use them as internal standards. They behave similar. They are chemically and physically the same, except that they are more heavy, and then there are smaller minor effects like you said, it could be some isotope effects with biosynthesis or. Yeah, maybe you can talk. Yeah. So I'll try to get the software started, see if it works. So you have the software it's very simple to use actually. So there's 3 tab sheets which can use. One is input, one is calculate, and one is look at the results. Now we have nothing loaded, so you cannot see anything. But input, you can say, for example, load settings. So if I know how I want to process, you can fill in the tab page of the calculate parameters here that has already optimized. So we do this now. We just load settings and see if we can find. So we take the DON example, and there is a setting file. I open this. And if you go here to calculate, then the parameter settings for the data processing is then loaded into this sheet already. So this says now we have we want to screen for metabolites or metabolization products which carry between 5 and 32 maximum carbon atoms. We inspect the chromatogram from 3 to 33 minutes. So it's not working, obviously, because I have not loaded the group. So what I'm doing next is I add groups. So the software is capable of processing the data batch-wise, but you could also have a group which is only carrying one sample file, so only one sample, and this can be done here. So we call the group, I call it talked on, and we try to add the files, and then we take, I don't know, this one is a processed file, so this is the MZ-XML file, which can be read by the software. We just take this. Base means that we took the whole ear, which we have analyzed of the wheat ear after treatment with Don. So we open that, and then it's listed here, and then we say OK, and then the data file is inspected if it carries all the features which are necessary for processing and all the MZ-XML characteristics. And if we go now to calculate, then we can enter here. And I say, because I know just to shorten a little bit, I lower the retention time window where we process the data to 20 minutes, and all the other parameters we leave it as it is. If you're interested in this, we can discuss this afterwards further. So we don't save, we don't need it at the moment. And this is all these things which are illustrated here are basically used to process single data files. And if you want to process whole batch, then you can here indicate this by ticking this mark. Then it's grouping the results and produces automatically this data matrix which I showed you in the morning. So we don't use this now. And then let's see if we can start the whole thing. Where it is. No, it's on the right. That's this one, because it's too okay. So do you want to save loaded data? No, we don't want to save something. Then the data file is now processed. First it's parsed. This takes a little bit, and now it's extracting these signal pairs. So meaning that it's looking for this matching non-labeled and fully labeled, and it indicates here that how the retention time progress is. So we set 20 minutes, so it's not taking very long, as you can see. It's almost done. It depends on how big the data files are. If you use the QTOF data with a 4 GHz mode of Agilent, for example, then it's a challenge for the program. This is it's working, but it's taking time then. And for the Orbitrap data, so if you have a batch, let's say, so you can process 50 samples without any problem overnight, for example. Yeah, for example. So that's a rough estimate, depending on the settings of the instrument as well. So I did not mention this now. So we are planning to finalize some things, and then we are going to publish it and make it available to everybody. Okay, so obviously it's finished. Then we look at the results, and if you go here, then you see it's extracted 1,000 M/Z signals from the whole data set, and this has been binned into 76 bins. This resulting in 58 feature pairs, which remain here, and this is grouped into 15 groups automatically now. So which has, of course, to be verified and inspected manually. But if you look here, now it's a bit of a pity that it's not very big, but here's the illustration of all the M/Z values. We can also have a look at the features at this screen. So this tells you now where are the features in the chromatogram. So we have the retention time here and have the M/Z, the intensity, now the M/Z value here. So look from top on the chromatogram again, and then you can indicate here what you want to have illustrated. So You just mark this or whatever you want. And if you look in the feature groups, then you see now that most of the feature groups only have one single feature. So there seems to be only one ion which has been assigned. And there's this feature group which I mentioned already. There's one with very many ions which are produced in of this yeah. Okay. But you can also then have a look at the mass spectrum. If you're interested in a particular feature group, we can, for example, take feature group 8 here. And then, let's say we start with the main. Then you see the overlay of all the ion features which are contained in this particular group. So all these ions have been assigned to the feature group 15. In total, it's 8. Retention time is 11.8 minutes. And here, this is illustrated as the overlay of the x-axis of all these ions attributed to this. And how the grouping worked and how the peak shape looks like, this can be then inspected here. So if we just zoom in and one peak, then you see this is how the data look like. Yeah, it's really perfect co-illusion, very same peak shape. And if you here say scale features, then everything is scaled to the most intense feature here. Then it's easier to compare the shape. But if you say, okay, we do not want to scale, then this is still not working properly. Christoph said he's going to fix that. So we have to first change the group again, go back. And then we can zoom in again. And then you see now the real relative intensities, how the chromatographic peaks look like, right? And but you also have a group overview which tells you the correlation. This is a correlation plot of all the features, how good they correlate. So it gives you an indication of if the grouping worked or not. There you can also have some problematic features which have been wrongly assigned, but this helps you to address that. You can just as a tool to verify. And you can also have a look at the mass spectrum. Then isotope logs are indicated if they have been found and used for the data for the evaluation. So if you go back again and go back here, then we have now the mass spectrum, and because it's all feature pairs. So if we zoom in again, we can take, I don't know, we just take this one. Then you see which of these have been really grouped together, right? Which form a pair. So here's one telling you here's the natural isotopic signature, and if you look at this, you can see this is the counterpart of C13. So this is how you can go walk through the data and have an idea of how it looks like. So, and what also is supported by the program is that if you want to export a tab separated file, or if you want to export a PDF, this can also be ticked at the beginning of the processing, and then for every feature group you get a PDF page where you have the summary of what has been processed so far. So yeah, I mean, this is maybe just to give you an idea of how efficient this data processing could look like, because it's just you push the button, then it's a minute, and then you have the whole data set which was measured in full scan mode and carrying these more than 10,000 features is reduced to that what you want to see due to the isotopic signature which you introduced by the metabolization experiment. And this is, I think, so I'm still very fascinated myself about the technology. If I'm talking about it, I always realize again how fascinating it is. So yeah, it gives you the chance to look into the data in a way which you would never have without that probe. Okay.